This is what we talked about yesterday. We looked at DNA replication. I did kind of a little example of how it could look using some of the DNA models that you guys made before. And then I wanted you to do this page, and that's basically all we had time for. So your version of this page here should have looked like this. And then your own words, a summary of DNA replication. Comments, questions, concerns on any of this? Is there anything anyone would like me to go over? Anything anyone would like me to explain or review? Going once, twice, thrice. Okay, cool. Sold. Let's make it more complicated. DNA replication and now protein synthesis. Find me this page. This is today's lesson. So DNA replication happens once. Your DNA gets in every cell. Your DNA gets copied, waiting for cell division. And in the in-between, protein synthesis happens a ton. Protein synthesis is how proteins get made. Your body's constantly using proteins, whether it's to make more of your hair or to tell your cells what to do or to make more of your blood or whatever. There's all sorts of proteins that your body is constantly in need of. And so this protein synthesis happens all the time. It's similar to DNA replication. But fundamentally different in a couple key ways. The process is similar. DNA gets unzipped. But there are some key differences to this. So here we go. After your DNA replicates, so you've got your cell has copied the DNA and now it's waiting for mitosis to start because the copying happens during interphase. After it replicates, now you've got chunks of DNA available to be used in your cell for a bunch of different things, mostly protein synthesis. That genetic code, your DNA, is copied in chunks. This is one of the fundamental differences between DNA replication and protein synthesis. In DNA replication, all of your DNA is copied. In protein synthesis, you only need to copy the pieces of your code that are responsible for specific proteins, and that's not all of it. So the genetic code is copied in chunks into a molecule called RNA. Now, you've seen this before. We've talked about it as being a nucleic acid. One of your questions was how are DNA and RNA similar, or how are they related to each other? This is another way that DNA and RNA are related to each other. RNA is what you get when you copy a small chunk of DNA to make a protein. Now, there are a couple key differences here with DNA and RNA that I'm going to outline for you. The first one is the name. Obviously, DNA and RNA start with a different letter. DNA starts with a D, RNA starts with an R. The D in DNA stood for what? Deoxyribose is what the D stood for. N is nucleic, A is acid. But that D stood for deoxyribose. The R in RNA stands for something similar. Does anyone want to take a guess what that R could stand for? It's related to a portion of the name for DNA. Ribo. Yes. Nucleic acid. RNA, the R in RNA stands for ribo. And that means there's, sorry? So what was deoxyribose? So deoxyribose is the sugar backbone in DNA. So RNA has a different sugar. Instead of deoxyribose, where there's no oxygen present in the sugar, ribo presumably does. It doesn't have the D. There is no deoxy label to it, so it's ribose sugar instead of deoxyribose sugar. It's oxygenated ribose sugar, I guess. That's how you want to think about it. Where am I here? There I am. So in ribonucleic acid, the sugar backbone is ribose. Ribose as the sugar backbone. Bless you. I'll zoom this out a little bit so you can see. There you go. So that's the first key difference. Why that different sugar? I don't know. I do know that DNA is a permanent molecule. It sticks around. Once it gets copied, it's there forever. RNA does not. RNA gets reused. Once it's been copied, it's turned into a protein. It gets resorbed by the body. Those pieces get used up other places. So the RNA shows up, and then the RNA gets translated. Wrong color. Translated into a protein. So it's not a copy, it's a translation, and there's a key difference there. I want to make a note here off to the side. So we've got DNA, and we've got RNA over here. First key difference is that sugar backbone, ribose instead of deoxyribose. The second key difference here is actually the nitrogenous bases that show up in RNA. 3 of the 4 that show up in DNA also show up in RNA, but there is one key difference. So to review, what are the nitrogenous bases that show up in DNA? Guanine, cytosine, adenine, and thymine. When you make a DNA strand, these are the nitrogenous bases that get used. RNA has 3 of these 4, but one key difference. I'm just going to use the letters for the ones that stay. G, C, and A, guanine, cytosine, adenine, they all stay. They are found in both molecules, but for whatever reason that I don't know the answer to, instead of thymine, when RNA gets made, everywhere where thymine should be, there is a different a different nitrogenous base that gets slid in there. And that nitrogenous base is called uracil. Instead of thymine, RNA contains a U. And that uracil gets placed exactly where a thymine would. So in RNA, uracil and adenine are the complementary bases, and cytosine and guanine are still the complementary bases to each other. So an analogy for protein synthesis, actually before I give you the analogy, I'll show you practically what it looks like. Where is what I did? Yes, this is the one I used. Okay, so here is a portion of your guys' DNA chain. Is this your class? Let's find out. It is. This is Lamise. No, the tape broke. Oh, I put you over it. Cut it somewhere. So when we were doing DNA replication, we took your DNA, the DNA chain that existed. The whole DNA chain gets torn in half, and then the individual pieces, the individual nitrogenous bases get tacked on the side. Hold it this way. Get tacked on the side, and you get 2 DNA molecules. Yes, I'm trying to demonstrate this for you guys. In protein synthesis, that doesn't happen. You don't copy the entire chain. You need the entire chain. You need a specific piece. So what ends up happening is that if this is our entire chromosome molecule, this big long chain of DNA, your body decides, you know what, I need to make more here. So I need to code for creatine. Sorry, keratin. Maybe the keratin code is over here. So what happens is this portion of DNA gets unzipped. Not all of it, just this specific piece that you need. And then just like in DNA replication, the piece that you need, from here all the way down to here perhaps, gets copied. The ribosomes get in there, and they're like, okay, for this guy I need this guy, for this guy I need this guy, for this piece I need this piece, for this piece I need this piece. And now this becomes your RNA that gets taken away. This gets stitched back together. It's just hydrogen bonds. They're attracted to each other on their own. The DNA zips back together. And then the DNA goes off and waits to get pulled apart and turned into a protein. Now, instead of, if this were RNA, instead of being C, C, T, C, G, T, C, A, C, A, C, we lost our garbage cans because the janitors don't want to clean them up anymore, so you got to go all the way up to the ones in the hallway. Sorry. The RNA code would be CC, and instead of a T, you'd have a U. It would be CCU, CGU, CACAC. And that's what your RNA piece would look like, and it would go off. So an analogy that I have for this, and there's a bunch of different ones. You can think of DNA as being a whole book. And let's specify here, let's call it consider it a rare book. A whole rare book. You're at a school, and your school has access to this rare book in its library, but because the book is so rare, nobody's actually allowed to take it out of the library. And so what you do is you're like, I need a piece of this book. I don't need all the book, I just need a piece of the book for a research paper that you're doing. Maybe it's English, you're in Mr. Sapine's class, you're learning how to write an essay on a particular topic, and the rare book in the library has the information, a chapter of the rare book in the library has the information that you need for this. So what you do is you get into the library. The book's not allowed to leave, so you bring your notepad. You're like, okay, it would be, I don't have enough time right now to copy out the entire chapter word for word. So what you do is you take your notepad and you make a bunch of notes. You read the chapter while the book stays in the library, and you make notes. And that's what that RNA is. The RNA is, it's a note form copy of a chapter. That says note form, like you've point form notes or bullet notes or however you want. You haven't written it down word for word, but you've got all the big ideas. That process has a name. That process is called transcription. And now you've got your copy, and then you're going to go back to your paper and you're going to turn that copy, your note form copy of the chapter, back into the chapter itself so that it's usable in your paper. You can reference it properly when you have the whole thing together. And so what the making of the protein is like, it's a rewriting of the chapter from your notes. And that process also has a name. It's called translation. You've taken your notes, you've turned them into something usable, the proper chapter itself again. And now this piece of information, the protein, can go off and perform its function. Maybe it's growing hair or your nails or your blood or whatever. So let's fill in some of these details. When needed, and this doesn't need, because your body doesn't need these proteins all the time. It only happens in specific instances. When needed, you need a particular protein. A cell accesses the genetic information coded within a single gene to create proteins for cellular activities. Now, there is a particular compound, it's a kind of compound that does this. It's called an enzyme. an enzyme called RNA polymerase, is used to copy the sequence of the DNA's nitrogenous bases to create a single strand of RNA nucleotides. This is what I tried to demonstrate for you here with your guys' molecule chain, your DNA chain. Once the RNA has been completed, or next, this single strand of RNA is used to produce the correct sequence of amino acids now we're to amino acids to build the protein. So the protein doesn't get built in one long strand, it gets built in chunks. The amino acid might only be 6 nitrogenous base pairs long, but the whole protein might be 24. So you slap 4 of these amino acids together, and now you've got your whole protein that can go off and fill a particular role. So it says here, thus the sequence of bases in DNA's genetic code will determine the sequence of bases on a strand of RNA. So it's a copy, which in turn will determine the sequence of amino acids needed to make a protein. We can see how it's similar. I'm hoping we can see how it's similar to, yet critically distinct from DNA replication. And I've got a picture of it down here on the bottom. So I'm going to zoom in on this. I'm going to make a note here make some notes on here for you guys. So here's an example of what protein synthesis would look like versus DNA replication. You can see here you've got your long chain of DNA, and for whatever reason, your body just needs a chunk of this. This object right here, this gray blob, is your RNA polymerase. Sorry? Yes, Malik, what's your question?
Okay, so basically he's asking that. So you know how like the hair requires protein to grow, right?
Yeah.
So what if like you? No, it's not. It's like if you don't have a protein in your hair, it's like fat.
No, this was actually a question that was asked in the other block, I believe. I've heard it once. I've heard this question once already, and I can't remember what block it was from. So ask the question.
So yesterday you said that like if you're hard on protein, it's gonna stop like protein from like the less important parts.
Yes, your body will redirect nutrients from less important parts to more important parts. Yes.
Yeah, like hair loss or like receding hairline, right? That's like protein.
No. So hair loss and receding hairlines happens as a result. I mean, your body's just getting old. Like it's like as you age, it perhaps isn't like there are certain parts that perhaps aren't able to function the way they were, and so you get balding happening. Why that happens earlier in some people than others, I don't know. The example that I've been hesitant to share. So what I really mean is when we're talking about like your body needs the nutrients in some places more than other places. The example that I always hesitate to bring up, but I will, is something like an eating disorder. So I don't know if you've ever known. I've had friends who have gone through this, but for people who experience an eating disorder, regardless of what it is, what the disorder is, ultimately what it results in is they're not getting enough nutrients in their body. So in like bulimia, you eat a lot and then you purge it, or in anorexia nervosa, you just don't take it in, you starve yourself. So if your body doesn't have access to the nutrients because you're getting rid of them all the time, or you're not eating properly, it could be just as simple as not eating properly. Like sailors back in the 1700s who were exploring the world, they got scurvy because they didn't have vitamin C. Is another example of this kind of thing. So it's when you don't have access to these nutrients, then the processes that go on to build them, your hair, your fingernails, are less important. Like in the grand scheme of things, do I have to have fingernails on all my fingers? Could I function if I took a hammer and smacked the end of each of my fingers, killed my nail beds, and didn't have nails on my fingers anymore? Other than the fact that that would hurt, would that negatively impact my life at all? Not really. But if you took a more critical protein, like, I don't know, the muscle that makes your diaphragm, or the protein that makes your heart muscle, if those things got inhibited in some way, well, your protein, your diaphragm doesn't work, you're not going to breathe, you're going to suffocate to death. If your heart can't beat, if any part of it is deficient in some way, now it's not pumping blood throughout your body, parts of you are going to start to die. And so it's the deterioration of things like that. So you can see one of the signs of an eating disorder, when you talk about it when I teach psychology, one of the signs of an eating disorder is, one of the first noticeable things, other than the weight loss, is really bad hair. Because it's not being built the way it's supposed. It will break, it will fall out in chunks. Like it's not being built properly. So that's what I mean by that, Malik. So not like a receding hairline.
Hey, another one. So I like split my head open before,
Yeah.
right?
No, let Malik say his things. Don't interrupt Malik. Namret.
And like, so the spot that I split my head open, like hair won't grow there.
Oh, interesting.
It's like,
So you've killed the follicles. So I had something similar happen when I was a child. When I think I was like 4 or 5 years old, I was gonna go say goodbye to my cousin. I really like this particular cousin, and his family. There was a bunch of kids in that family. I wanted to say goodbye to my cousin Kevin. So I shoved my head in, and being a big family, they had a GMC Astro van. Those of you, it's a large van. And back in the day, the vans had didn't have automated sliding doors. You wanted to close the door, you put your hand on the door, and you shove really hard. So my oldest cousin, Jeremy, Kevin's older brother, was in the back seat, and he didn't know that I was coming up beside the van to say goodbye to my cousin Kevin. And so he slams the door. I stick my head in the door to say goodbye to Kevin, and I took the GMC van door right here. There was no blood. I'm fine. But, and students will point this out periodically, if I'm sitting at a desk helping someone and another student comes over every so often, usually happens about once or twice a semester, the student will be like, Mr. DeWita, do you have a circle of gray hair or a circle of white hair? And I do. Back here, if you stand over me, you can see it. I've got a chunk, probably the size of a quarter. It blends in. Normally, it blends in well with the rest of my hair, but when my hair is longer like it is now, you can see it a lot easier. I've got a chunk of gray that I've had since I was about 5 years old when I got my head slammed in the van door. And it's not, those follicles didn't die entirely, but the portions of the cells that produced color in my hair did. And so I've had a patch of gray since I was 5. And so like there's different kinds of damage that can happen to different kinds of cells in your body. So you have a patch with nothing. You got a whole lot more hurt than I did. But it's unrelated to this process. So back to this process, protein synthesis. You've got the RNA polymerase, and what the polymerase does is it starts at the beginning. Whatever the beginning is, there's a portion of the code where it's like, yeah, this is where we need to start. And when the DNA unwinds, there's 2 strands. One called, and again, you don't need to know this vocabulary, but I'm going to expose you to it. There's an antisense strand and a sense strand. And it's the sense strand is the actual, this side is the side, the antisense side is the side that would actually code for the protein. So it's the side that gets copied by the polymerase. So you can look here where I've got CTG, and the way this thing gets copied is it gets copied in 3 nucleide lettered chunks. Those things are called codons. Now, if you've got friends in Mr. O or Mrs. Broad's class, Miss Broad's class, they go into a lot more detail about how this happens. I'm choosing not to, because it's way beyond the curriculum, and there's other stuff I'd rather cover with you. But I know they go into a lot of detail. There's whole charts and that they, there's whole charts that they go over that I'm not going to. But simplified, this RNA polymerase is going to read it in 3 letter chunks. So it's going to say, okay, CTG is going to become, well, C goes with G, T goes with A, G goes with C. We're good. Now the next three-letter chunk goes ACG. Now A would normally pair with T, and you can see it down here that in the DNA, A right here pairs with T down here, but when you're building RNA, thymine doesn't get used. So wherever thymine would be placed, that's where the uracil goes in. So this is the RNA piece that's getting built, and you can see where A should have paired with T, A gets paired with U instead. So then the code goes U, C pairs with G, G pairs with C, and those three-letter chunks are done. And then you get the next three-letter chunks, GAT, G goes with C, A goes with U instead of T, gentlemen, in the middle. T goes with A, and you go all the way along until somewhere in the code there is a signal for the RNA polymerase to be done. It stops copying, and now this RNA transcript gets pulled out of the code. These guys hydrogen bond back together, and then this RNA will go off into another portion of the cell and get copied to be a protein again. So let's do an example of this for you guys. This ATG is the code to start making this RNA transcript. So if this is what we're starting with, let's go ahead and make the RNA from this. I'm going to do a couple of these, and then I'm going to get you to finish it. So if the DNA chunk starts with ATG, what should the RNA start with? Well, in the RNA, A is going to pair with what? U. So right off the bat, that first letter should have been thymine in the DNA, but in RNA it turns into a U. Now in the next letter in the DNA is a T, so in the RNA it's going to be an no, T pairs with A, and A is a nitrogenous base allowed in RNA, and G is going to pair with C. Cool. So the first three letters of the RNA are going to be UAC instead of TAC. And now in the second three, it goes ACG in the DNA, so the RNA is going to go. A is going to become paired with U, C is going to get paired with G, and G is going to get paired with C. So when ATG, ACG in the DNA, in the RNA goes UAC, UGC. What are the next three letters going to be? It's going to be CUA. And then the next three letters are going to be you betcha, GUC. I think Josiah, that was your voice. So we've gotten to here. Oh, that was Luca's voice? So these 1, 2, 3, 4 chunks represent these 1, 2, 3, 4 chunks, and we've started building our DNA. So we've started building our RNA. And now this is going to continue. From this stage, this guy here is this guy here. I want you to finish building the RNA code. And this is as far as I'm going to take you guys today. Finish the RNA code. And then I want you guys, there's a whole set of questions I want you to play with. All the way up to, in your notes, you're going to see something here that says heredity denotes genetics activity. I want you to stop on the page before that. You're going to go all the way up and finish up to page 20 or question number 24 on page 14. You do not have to do question number 25. You're going to spend the rest of the class, there's about 40 minutes left, working through DNA replication and protein synthesis. You guys are going to have a quiz this week on DNA structure, function, and protein DNA replication and protein synthesis, just so you're aware. Okay, starting here on your mark and set giver. Let me know how I can help you. Sorry?
The quiz example is about.
I have not said that. I said before the end of day Friday, you will have a quiz. It might be tomorrow. Might be Thursday. Might be Friday because I'm not here Friday.
No, it's not Friday.
I don't know, days I'm not here are usually really good days to give out quizzes. But who knows?
He's lying.
Okay. Yes, Malik. You've got your task. Keep going.